Hi everyone, Ethan here. Today I'm going to talk about nonsense-mediated mRNA decay. We're covering genetics in class. I figured it'd be a good idea to cover it. But I know what you might be thinking. Ethan, of all the topics in genetics, and there are many, you chose that? Why? The answer is simple and stupid. My eyes were just simply drawn to one word, and that's nonsense. As well as you all very well know, first off, most of what I say is nonsense. But second off, I happen to really like saying the word nonsense. I'm like a child. I get fixated onto words and I just repeat them over and over and over again. So when we covered this idea in this concept in lecture, I saw the word nonsense. I was immediately drawn in. And once in, I was like, oh, geez, wow, this actually covers a lot of important genetics concepts. So I figured, yeah, let's make a video on it. So before we talk about this decay pathway, I want to talk about what it targets, which are nonsense mutations. What is a nonsense mutation? Nonsense mutation is pretty simple. Recall that in transcription, you got RNA polymerase on DNA creating an mRNA transcript. And sometimes these RNA polymerase makes mistakes. Sometimes it substitutes bases when it shouldn't have. Usually, or I shouldn't say usually, many times this doesn't actually matter, because remember that the codon code is degenerate, meaning that there is some redundancy even if an error is made. But in the unfortunate case of a nonsense mutation, this error does matter, and you end up substituting one base, which causes substitution of an amino acid for a stop codon. So that's not good. But it doesn't actually matter at this point. You still create the whole transcript, and that transcript does actually go to the cytoplasm, and everything's, fine, good, and dandy. It actually goes all the way to the ribosome. And this transcript does, in fact, get translated. In this translation process, though, as, the ribosome's going down, do, creating its little, polypeptide chain, eventually it's going to hit that premature stop. And what does it do? Well, it does what it's told. It stops prematurely. It terminates translation. This sort of nascent polypeptide is attached and it, releases from the mRNA. But remember, it's premature. There's still a lot of mRNA. It was still supposed to translate, and instead we're stopping there. So the protein product, or the polypeptide product that's created from this translation process, is fundamentally incorrect, and it can be catastrophically incorrect, right? You could have stopped like 1/3 of the way through, and there could have been 2/3 of it left, and that 2/3 could have had, the active site for an enzyme that was in this protein product, and it could be disastrous. And the way I sort of think about it is like that nightmare scenario when you're taking a test. When you're taking a test and you finish really, really fast and before everyone else, you think, oh, I'm hot stuff. I'm ready to go check everything. I'm done. So you turn it in early. And then after the fact, you're talking to your friends and you realize, oh boy, oh no, there was like a whole backside you didn't cover. I finished too early. I finished prematurely. It's a premature stop, just like a nonsense mutation. Premature stop that can be catastrophic for your test, also can be catastrophic for your cell, right? Because you're creating this wrong protein product. So, okay, that's very bad. That's clear. Nonsense mutation. Okay, that's clear, Ethan. How does the mRNA decay pathway work? How does it combat this problem? Well, it starts working way before you even get to the translation step. Recall that there are many intermediate steps between transcription and translation, right? And one of those key intermediate steps is splicing. Splicing is when you have the mRNA transcript, we're going to take away these non-coding introns, so you only have these coding exons left. And in reality, in the splicing process, these things called exon junction complexes are placed. This is the exon junction complex. Do I want to take anything away from this picture? No. I mean, I'm not smart enough to take anything away from this picture, but this is what the protein complex looks like. This protein complex, as sort of the name tells you, is like a junction between these exons. So recall, if this is your exon, intron, exon, pre-mRNA transcript, and then pre-splicing transcript, then you splice, the EJC is placed to join two exons. And I know my penmanship's horrible, my drawing is horrible, but hopefully this is clear enough, right? This EJC, exon junction complex, sits between and joins two exons that were once bridged by an intron. And sort of this mRNA transcript that is studded with EJCs, goes in the cytoplasm and ends up getting translated. And during translation, during the whole translation process, as the ribosome moves down the mRNA transcript, I like to think of it as a bowling ball and bowling pins, right? As it moves down the transcripts and encounters EJCs, it just knocks them off. And it just keeps going down, boom, 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 knock off, knock off, knock off, knock off. And you can think if there's no mistakes, if everything goes fine and dandy, the ribosome is going to go down the entire transcript until it hits a stop codon, and that's going to encompass all of the exons, meaning it's going to knock off all of the EJCs, so that when that ribosome finally detaches from the mRNA, there should be no EJCs left. And that's sort of the fundamental basis from which nonsense-mediated mRNA decay works. Because ideally, there should be no EJCs left. But if you think about it, there was a nonsense mutation, there would be EJCs left. Why? Well, a premature stop codon means what? You're going to stop, and the translation is going to stop. The ribosome is going to detach prematurely. They're still going to be coding exons downstream of that stop with, as a result, not just coding exons, EJCs. So this is sort of the fundamental mechanisms for how this pathway works. So remember, if there's a nonsense mutation, the ribosome encounters a stop, there are release factors, ribosome detaches from mRNA, there's still going to be EJCs remaining, right? And this remaining EJC is the key in nonsense-mediated mRNA decay. After termination, and this is true whether or not you have a nonsense mutation, a protein complex is kind of formed with UPF, right? UPF is one of those things. And UPF has SMG also joined. This is like a new protein complex at termination. UPF1 ends up recognizing UPF2 and UPF3 as a part of that EJC. And it basically says, oh boy, we messed up. And a lot of proteins come in, SMG proteins 1, 8, 9. I was reading some papers on it, and they're not fully sure actually. There's just a lot of SMG proteins. It is well beyond my comprehension. So thereby, I don't want to explain things wrongly. Just know that a lot of proteins are involved. In addition to this basic complex, a lot more SMG proteins and other proteins are involved. And there's a complex mechanism that's multi-part. There's going to be like decapping of this mRNA transcript. There's going to be exonuclease activity. Basically, the final end result is you end up destroying this mRNA transcript. And that's really good because it thereby prevents additional translation of this faulty mutated mRNA transcript with a nonsense mutation. So again, do not quote me on all these proteins. I just know that UPF1 is part of that termination protein complex, recognizing UPF2, UPF3, and then a lot of SMG proteins, other proteins come in, decap exonuclease, basically end up destroying this mRNA transcript. And that's good. So you don't end up having more mistranslations. But this was a question I had. You might be wondering, okay, Ethan, you destroyed the mRNA, but what about that nascent polypeptide that is now floating around that was, stopped because you hit the stop codon and attached and it's just floating around. And what happens to that? Turns out that I read this paper that there is a protein quality control mechanism. There's some like ubiquitination, some proteolysis that occurs. I'm going to be honest with you, I didn't fully understand this paper. And I think that the author even notes that it's not as well studied as the mRNA decay pathway, but there is some protein quality control that's coupled with the nonsense-mediated mRNA decay, which is cool, right? Because you don't want this, faulty protein circling around, and it's cool the cell has a way to deal with that too. But that is beyond the scope of my knowledge, well beyond the scope of my knowledge, and therefore well beyond the scope of this video. So just a review, that's basically, well, to be clear, that's nonsense-mediated decay in short. Obviously, there's a lot more detail. If you read papers about it, you'll say, Ethan, well, you missed this, you missed this, you missed this. But that's the idea in short. As a review, what is a nonsense mutation? It's a premature stop codon created during transcription. And recall that post-transcriptional slicing leads to the placement of these exon junction complexes. So this nonsense-mediated mRNA decay involves the recognition of remaining EJCs post-translation, because usually the EJCs are detached during translation by the ribosome. So these remaining EJCs after the termination of translation are recognized and therefore initiate mRNA breakdown by that complex of UPF, SMG, all that stuff, does an exonuclease, all that stuff to destroy the mRNA that is faulty. So that is my short explanation of nonsense-mediated mRNA decay. Here are my sources. as always, I'm glad this lecture was shorter than the last. If you have any corrections, advice, criticisms, feedback, anything really, feel free to leave a comment. Let me know. Hope you enjoyed it. Hope you learned something. If not, also let me know, because I want to make sure you learn at least a little bit through these. But then you may be an expert, not to learn anything at all. Which for that case, while you're watching this, I don't know why I'm doing now. This is wasting your time. In short, thank you all for watching. Hope you are well. Take care. Bye-bye.